Afternoon. My name is Jeff Meacham. I'm the Senior Biopharma Analyst here at Barclays. We are thrilled to have CytomX Therapeutics present this afternoon, and speaking on behalf of CytomX is Chairman and CEO Sean McCarthy. Sean?
Great. Thank you very much, Jeff, and good afternoon, everybody. Pleasure to be here to present an update on our company. So as you'll hear over the next 24 minutes or so, what we're doing at CytomX is really rethinking the therapeutic antibody, initially for the treatment of cancer, rethinking and, if you like, reimagining therapeutic antibodies. Over the course of the next few minutes, I will be making certain forward-looking statements. I refer you to our regulatory filings on our website. So, of course, we all know that therapeutic antibodies are a very successful antibody therapeutic class. These are powerful and potent modalities, generating more than $100 billion of sales in 2018. However, we're also learning increasingly that the potency of these agents can also be a liability, in that where targets for therapeutic antibodies are widely distributed, this can lead to narrow therapeutic window and to on-target toxicities. So we believe there's a major opportunity to improve targeting and localize antibody pharmacology in a number of areas, including particularly in oncology. So we're doing this with a very unique technology platform we call Probodies. Probodies are fully recombinant antibody prodrugs. They are designed to be active selectively within the tumor microenvironment. And the way we achieve this is by engineering the antibody to contain a mask. The mask blocks the ability of the antibody to bind to target until the mask is removed selectively and specifically in tumor tissue by proteases, which are enzymes resident in cancer tissue. This allows us to fundamentally alter the distribution of antibody into tumor tissue and away from normal tissues. The company is based on more than a decade of deep scientific research, including deep protein engineering experience and intellectual property, and also a deep understanding of the tumor microenvironment. Over the last couple of years, our therapeutic pipeline has come into great shape and really come into focus. We have two wholly owned lead programs, which I will describe to you today. The first is CX-072. This is an anti-PD-L1 Probody, which we believe has the potential to become a differentiated centerpiece of combination cancer therapy moving forward. The second wholly owned program is CX-2009. This is a very different molecule. This is what we call a Probody drug conjugate targeting a first-in-class cancer target called CD166. We also have two collaboration programs in the clinic. First of all, BMS-986249, which is a Probody version of ipilimumab, Yervoy, the anti-CTLA-4 binding antibody. That's in a phase 1/2 study being run by BMS. And the second partner program we call CX-2029. This is a Probody drug conjugate targeting another very unique tumor antigen called CD71. So again, the pipeline has really taken shape recently, 4 programs in the clinic, and I'm going to share with you today an update on data on CX-072 and some preliminary phase 1 data that we released a couple of weeks ago on CX-2009. So CX-072 we see as a differentiated anti-PD-L1 Probody therapeutic. So I should stress at the outset, this is not an antibody, it's a Probody. Probodies have very unique clinical pharmacology, and this, we believe, has the potential to be a differentiated addition to the immuno-oncology landscape. Our target product profile for this asset is for CX-072 to potentially become the centerpiece of combination cancer therapy moving forward. And our basic concept is that immunotherapy is associated with a wide range of immune-related adverse events and side effects. By using a Probody that's more localized into tumor tissue, we can more effectively and more aggressively combine with other agents to get more patients into long-term durable remission. That's our overall goal. And over time, we will be combining this agent with multiple different mechanisms, enabling more effective combinations. And so the underlying safety improvement inherent in Probodies, we're leveraging to get to greater levels of efficacy. That's the basic idea. So we completed dose escalation last year for CX-072 in the all-comer setting. We escalated from 0.1 to 30 milligrams per kilogram. We actually did not reach an MTD, but the drug was well tolerated, and we selected 10 mg per kg as our dose for expansions. In the course of this dose escalation, we learned some very important things about the platform. We checked some really crucial boxes at the molecular level for the first time, showing how probodies function in cancer patients. First of all, it's really crucial that in cancer patients, the mask stays on the antibody in circulation. Remember that the mask is tethered to the antibody via a protease cleavable linker. So it's important that the design that we optimized preclinically would translate into the clinic, showing that the mask is stable and that the prodrug is stable in the blood of cancer patients. And this box was firmly checked by our pharmacokinetic data. Secondly, we've been able to do direct measurements of probody activation in tumor tissue. We've developed assays to show that the mask can be specifically and selectively removed within tumor biopsies. We also have demonstrated, as I'll show you, very clear evidence that the prodrug can be active in a wide range of different cancer types. And we have a strong trend towards improved safety, both as a monotherapy and in combination with Yervoy. So this data has translated very nicely from our preclinical studies into the clinic. This molecule clearly has very unique molecular and clinical pharmacology and is the first example of an entirely new paradigm for therapeutic antibodies. The power of the platform is further evidenced by this data, which is fairly recent from Elizabeth de Vries lab in the Netherlands. This is imaging of a patient with an anaplastic medullary thyroid carcinoma with zirconium-labeled CX-072. And you can see that the probody lights up not only the initial tumor, but also a wide range of metastases. So this is direct evidence in the clinical setting that the probody, remember when it's administered as the prodrug, it can't bind target until the mask is removed. This is direct evidence using clinical imaging that the prodrug can be activated in primary and metastatic tumor settings, bind to target, and I'm pleased to say elicit a therapeutic effect. I'll show you on the next slide that this patient had a deep response to the drug candidate. So the platform is performing in cancer patients exactly as we had designed it in our preclinical studies, which is extremely exciting. This is a waterfall plot showing the anti-cancer activity of CX-072 in phase 1 dose escalation at doses of 3 milligrams per kilogram and above. And you can see clear evidence of anti-cancer activity. That patient I just showed you with the thyroid cancer is on the far right of this plot, so you can see the depth of the response. We've seen activity in a range of different tumor types, and I should stress that this is a phase 1 all-comer, heavily pretreated patient population, not necessarily expected to respond to single-agent PD-L1, so encouraging evidence of anti-cancer activity from this phase 1 dose escalation. Encouraged by our phase 1 work, as I mentioned, we picked a dose, 10 milligrams per kilogram, as our expansion dose, and we've been expanding now in 8 different tumor types over the last several months. What we're doing is we're enrolling 15 to 20 patients in 8 different histologies to continue to understand the efficacy fingerprint of this very novel agent across a range of different tumor types, some of which we know are responsive to PD agents, some of which are a little bit more exploratory. I'm going to show you just initial data on triple-negative breast cancer, unspecified pleomorphic sarcoma, squamous cell carcinoma, and anal squamous cell carcinoma on the next slide. These expansions are ongoing. This is an early snapshot of the first 4 indications that we've studied. And again, you can see across these indications clear evidence at 10 milligrams per kilogram of anti-cancer activity in a range of different tumor types, which is encouraging and continues to show us that across a range of different tumors, the tumor microenvironment has the ability to remove the mask and turn the prodrug into the active species, resulting in anti-cancer activity. Just a couple of case studies on what this molecule can do in late-stage cancer patients. These are 2 triple-negative breast cancer patients, both of which have been very heavily pretreated, both of which, interestingly, had significant skin metastases, which is something that occurs in 15 to 20% of triple-negative breast cancer patients. And you can see that not only did we see responses in visceral lesions in response to administration of CX-072, but we also saw effectively complete responses in the skin to this widely metastatic disease in these patients that really had reached the end of the road in terms of available therapy and had been through many, many different cycles of therapy prior to coming onto our drug. So very encouraging activity in TNBC as a single agent. Similarly, in cutaneous squamous cell carcinoma, this is the indication that Regeneron's anti-PD-1 antibody has been approved in. You can see, again, powerful activity of the drug in this patient who, again, had exhausted all treatment options and was brought onto PD-L1 Probody. And you can see the dramatic response in this rather substantial scalp lesion that this patient had. So very clear evidence that the pro-drug antibody can be effective in a number of different tumor types, which is exactly what we wanted to see. How about safety? We've demonstrated very clearly through pharmacokinetic measurements that the prodrug is remaining masked in the circulation of cancer patients. What does that mean in terms of overall safety of the drug candidate? Well, it looks encouraging. Last year at ASCO and ESMO, we didn't really have enough patient data to really say anything about safety, other than that the drug was well tolerated. But as we get more experience with the drug candidate, and here I'm showing you 50 patients treated with 10 milligrams per kilogram, I want to highlight the incidence of treatment-related grade 3-4 adverse events, which is 4%. This compares very well to the generally accepted number of about 15% grade 3-4 treatment-related adverse events in PD-1 and PD-L1 monotherapy, and that's been well established, including in a recently published meta-analysis of more than 10,000 patients. So the monotherapy is trending towards being a safer molecule, is clearly an effective molecule, and the overall clinical pharmacology really is beginning to fit together quite nicely. So let's move now to, I'm actually going to skip this slide and move to the IPI combination. Again, just to remind you, what we're trying to do here with this prodrug PD-L1 antibody is have a safer, more effective centerpiece for combination with other agents. And the combination that we've studied most deeply so far is with ipilimumab. Now, it's well established that when we add PD-1 or PD-L1 to CTLA-4, efficacy, of course, improves quite significantly, but also, unfortunately, incidence of grade 3-4 treatment-related adverse events also increases significantly. There seems to be a synergy of toxicity between these two mechanisms. The data showed here on the left-hand table is highlighted in the boxes is the single-agent incidence of grade 3-4 adverse events for patients treated, melanoma patients treated with ipilimumab alone, 27%, and treated with IPI and Nivo together, 55%. So I'm going to come back to those numbers a bit later on. What we're trying to do with the PD-L1 Probody is see if in combination we can diminish the incidence of toxicity but maintain efficacy by keeping patients on full dose IPI for longer. So we initiated a dose escalation study where we escalated PD-L1 Probody CX-072 at 3 mg per kg of IPI, dosed every 3 weeks for 4 cycles. That's the full labeled dose of Yervoy, which is not used that much. As you know, in many clinical studies over the last several years, the approach with IPI has been to back off the dose, either to lengthen duration of dosing or to drop the dose altogether. For example, Checkmate 227, where the dose of IPI in combination with Nivo was dropped to 1 mg per kg every 12 weeks. So we're giving patients full dose Yervoy, and the results are really very encouraging. So this is a spider plot showing the durability of responses, again, in late-stage cancer patients that are not expected to respond to the combination. And you can see a number of deep and durable responses. We have a response rate of about 25% in this patient population at various doses of the PD-L1 Probody. And some of these patients have been on the PD-L1 Probody now for more than a year. And it's this deep durability that we're looking for, we're looking to capitalize on with Yervoy at full dose. So how about safety? Well, like with the monotherapy, the safety trends are looking very encouraging. So far, in 27 patients dosed with this combination, we see an incidence of grade 3-4 adverse events, which is 25%. So that compares very well to Yervoy monotherapy. That's also recapitulated at the 10 mg per kg plus 3 mg per kg dose, where we have 8 patients here with a 25% incidence of grade 3-4 adverse events. So the safety is moving in the right direction. It looks like we've been able to improve the tolerability of full dose Yervoy over 4 cycles by administering in combination with our PD-L1 Probody. And now we're in the process of developing next steps for expansion phase to go into a number of select tumor types to really firm up both the response rate and also the overall safety profile. So in summary, for CX-072, an emerging product profile consistent with our Probody platform vision. Single agent clearly has anti-cancer activity. We have a differentiated monotherapy and combination safety profile. And our next steps will be completion of these monotherapy expansions and initiation of expansions with the IPI combo in select tumor types in the coming months. So let me switch gears now to a very different product candidate, which is called CX-2009. This is, we think, a really big idea, something that we don't think could be done without our platform, which is to target a very unique tumor antigen called CD166, which is one of the most highly and abundantly expressed tumor antigens that we know of. No one's tried to drug this target before because it's present on most normal tissues at very high levels. It breaks all the rules of being a typical ADC target, but we think it's a very attractive target, and we've tried to drug it using our technology. So CD166 is a highly expressed cell surface adhesion molecule. Its biological function is not well understood, but it's present on most solid tumors, including breast, lung, ovarian, head and neck, and is considered to be undruggable with conventional approaches due to expression on normal tissues. So we believe the Probody platform enables the potential development of this attractive target with CX-2009. CX-2009 is a masked antibody against CD166 conjugated to the immunogen payload DM4. So our goal is to limit binding to normal tissues by masking, but to have anti-cancer activity as the mask is removed in the tumor, allowing the payload to kill tumor cells. We recently presented our first data from dose escalation, phase 1 dose escalation for this very unique experimental agent. We dose escalated in 7 tumor types. These 7 tumor types were selected for their high-level expression of the target. We escalated from 0.25 to 10 milligrams per kilogram, and we had 2 different arms, a part A, which was unselected patients, and A2, in which patients were selected for high-level expression of the target. Of course, the principal objective of this dose escalation was safety, particularly given how broadly expressed this antigen is in normal tissues. Can we dose escalate safely without any obvious on-target toxicity in normal tissues was question number 1. And I'll come to the safety in just a moment. Of course, what we're always looking for in a phase 1 dose escalation with a novel anti-cancer agent is any evidence of anti-cancer activity. I'm pleased to say that we did see some very encouraging evidence of single-agent activity in this study. This is a waterfall plot showing at doses of 4 mg per kg and above a number of responses to the agent. Again, this is a very heavily pretreated patient population, median of 6 prior regimens, but some very encouraging clinical activity, including here a patient with triple-negative breast cancer that has metastasized into the chest wall. And you can see very robust responses to the drug candidate. Again, this is single-agent CX-2009. And this patient is particularly interesting because she had had 4 prior regimens, and the two regimens immediately prior to coming on to CX-2009 were Pembro plus paclitaxel, which she failed, and then the Immunomedics TROP2 ADC, which she also progressed on. So this patient has really come to the end of the road with very aggressive metastatic disease, and she responded very effectively to our drug candidate. So exactly what we wanted to see in dose escalation, and really beginning to set the stage for further expansion phases and phase 2. We also measured directly in the blood of cancer patients the presence of the mask on the Probody, which again, as I've mentioned, is really a crucial success factor for a drug candidate like this, given how broadly expressed this antigen is on normal tissues. And these PK curves show the total and intact amounts of CX-2009 at various doses in the phase 1 study. First of all, you can see very nice linear PK, and also that the mask, given that there's no difference between these 2 lines, it's evidence that the mask is staying on, and this is important for what I'm going to show you on the next slide in terms of the overall safety profile. The drug was generally well tolerated at doses of up to 10 milligrams per kilogram. We actually did not formally hit an MTD. The principal toxicity that we're going to be managing moving forward is the expected ocular toxicity from the DM4 payload. We know that when you get to certain doses of 6 to 7 mg per kg and above, patients treated with DM4 conjugates do, if they're not pre-treated with ocular prophylaxis, they do demonstrate blurred vision and ocular toxicities. We know how to treat this. This is treated preemptively and prophylactically with steroid eye drops. We didn't prophylax in phase 1 because we wanted to understand the full range of the safety events. And as we move now into phase 2, that will be obviously something we'll be doing to maximize duration of dosing for patients. We saw 5 responses in dose escalation. Those responses did not confirm due to patients, for the most part, coming off drug for ocular toxicities at the higher doses. But we have every reason to believe now, as we institute preemptive ocular toxicity management, that we'll be able to keep patients on drug for longer and really get a handle on what the overall response rate is for this very experimental agent. So next steps for the CX-2009, we're doing some additional dose ranging now with ocular prophylaxis to select our phase 2 dose, and we will be initiating phase 2 expansions in several tumor types as the year progresses. So we're very pleased with our pipeline progress. The 2 lead programs, which are the first probodies to go into the clinic, are both showing encouraging activity, encouraging safety profiles. They're behaving as we've designed them. Both of those molecules are wholly owned. Just to reiterate that, we have the 2 additional partner programs with BMS and the AbbVie partnered CX-2029, where we expect to make additional progress this year, and then a tremendous amount of additional innovation in the laboratory that I don't have time to talk about today. A word about our alliances. We have been very successful in forming pharma partnerships over the years. Our foundational alliance with BMS, first struck in 2014, is in full force and effect. The leading edge of that alliance is our anti-CTLA-4 probody, BMS 986249. So far, we've brought $287 million into the company from BMS in this alliance. This was initially a 12-target deal. A few of those targets have been deprioritized by BMS, but the collaboration is still very active, and they are guiding towards an initial update on the CTLA-4 probody sometime this year. Our alliance with AbbVie is focused, the leading edge of that alliance is our CD71 probody drug conjugate called CX-2029. There's a number of other targets active in that collaboration. We have global co-development rights on that asset, and we're running the clinical program. And then we have a collaboration with Amgen in the field of masked T-cell engaging bispecifics. This is a very exciting area that we think has the potential to bring T-cell engaging bispecific technology into the realm of solid tumors. And we presented preclinical data on an EGFR CD3 T-cell bispecific, which we're very encouraged by. And that's the leading edge of our collaboration with Amgen. To date, from our partnerships, we've brought in about $400 million of capital. These deals have been not only a way to take additional shots on goal with the technology, but also an important source of non-dilutive funding. And taken together, our equity-based financings plus our collaboration financings have put us in a very strong cash position. We finished 2018 with $436 million on the balance sheet, which funds us comfortably into 2021 to continue to advance this deep and differentiated pipeline of probody therapeutics. So in terms of what to look for next from the company, for the CX-072 probody, we'll be providing additional guidance on specific milestones for the monotherapy. We're wrapping up the monotherapy expansions as the year progresses. We'll be announcing our strategy for the combination with IPI in terms of tumor types and expansion strategies, and also presenting an update on another combination we've been running over the last 18 months, which is with the BRAF inhibitor Zelboraf. For CX-2009, the CD166 probody drug conjugate, we are, as I said, doing some final dose finding to pick our phase 2 dose. We'll be announcing our specific strategy on indications and dose in the coming months. And as I mentioned earlier, BMS anticipates data disclosures for the CTLA-4 probody sometime during 2019. So companies making terrific progress. We're very excited about the technology, the pipeline, and our partnerships. And I'm happy to take questions in the breakout session, which I believe is in the New Yorker room. So thank you very much for your time.